So on Tuesday, recall we talked about like how you want to select your cells. Today we're going to be talking about some methods that are more specific to analyzing at the cell level. At the end, I'll also touch upon the fact that like many of the methods that we've talked about before, like using fluorescence, using mass spec, using NMR to extract things from cells and the like, can be applied to the cellular level and not just the molecular level as well. But we'll talk about 2 or 3 more cell-specific methods, specifically flow cytometry, and also some mechanical methods of looking at forces, traction force microscopy, micropipette tips that are specific for at the cell level. And when I'm categorizing cell type techniques and methods, I kind of split them up or categorize them in my head about doing something about at the population level versus the individual cell level. So for the individual cell level, we touched upon this on Tuesday that if you can study under unicellular conditions, you can remove the heterogeneity. That's present in tissues or multicellular environments. In like the same light as when we talked about single molecule techniques. Also, this is like a test tube for the fundamental unit of life. So those are why you might pick to focus on a unicellular condition versus if you want to do population studies, which I have like these images here, that if you want to do cell populations, that's where you can get at like how are cells interacting with one another. And cell population studies also have a lot of work in, okay, I just mentioned here, tissues, biofilms, depending on what type of cells you're interested in. Biofilms would be more on the bacteria, prokaryotic side, tissues would be more on the eukaryotic side, or if you're doing human relevant work. But if you're looking at a population of cells overall, a lot of these studies are looking at how evolution or fitness plays a role over a large group of cells. So this might be understanding like how well do cells survive based on like, let's say their population density. Or if you introduce some drug, how do they develop resistance? Antibiotic resistance is a big problem. One of our first speakers for the biophysics colloquium series, Kevin Wood at University of Michigan, he is really focused on antibiotic resistance. And I think, Julia, you're working at the clinic, right? You guys do cell?
In Jake Scott's lab, and our postdoc actually is from Kevin Wood's lab. He just joined our lab. So.
Okay. Yeah, so Julia can provide us lots of info about like thinking on the evolution scale of how cell populations change upon exposure to certain external factors. And even cell populations, you'll also see studies focusing on synchronization. So how do they, again, interact, work with one another? If one cell makes a change, does it influence its neighbors? Does this change over time? Those are some of the, I guess, areas of research in cell population studies. And many of the techniques like this image here, this was from Kevin Wood's work, you can see ampicillin, different doses, and they were looking at like how many cells die versus survive, where fluorescent indicators are telling you about the state of those cells. So again, many of the methods we've talked about before, the microscopy, optical techniques, are then used as readouts as well. But trying to get at either cell populations or single cells, a common tool that's used that I want to focus on that's, to my understanding, mostly used at the cell level, maybe some vesicles, as well as flow cytometry, where you're counting or trying to identify the number of cells within a population, analyzing them one at a time, and based on some optical readout will tell you about the state of those cells. So how this works is that you have single cells in suspension. So remember, we talked about suspension on Tuesday's lecture. So they're in solution, they're not adhered to a surface. If you are culturing your cells on a plate, you might have to remove them and then put them in solution or suspension, and they're going to be simply passed through a field of illumination. Before doing this, the cells themselves are going to be, you can stain, label them with fluorophores. That are indicating a specific molecule or specific state of the cells. And you'll then detect fluorescence and scattering from these cells. So, and the amount of fluorescent signal or the amount of scattering will tell you some aspect of the state of those cells. So you can see a schematic over here where this portion is, I guess, our source is a laser. Our sample is going to be the suspension of cells passing through a nozzle. So this is going to be a flow cell that passes the cells quite quickly. So it forms a liquid jet that moves at like 5 to 10 meters per second. It's a fast flow. That flow will be, and this microfluidics, sometimes it's a microfluidic technique, will isolate individual cells in that stream. And then, so that's what's passing through here. The laser light will then hit that cell. The light will interact with the cell, and you can see that you have, for the detection, that's all of this guy right here, is a fluorimeter or spectrometer type setup. So it has all of the dichroic mirrors, filters that we've talked about back in the fluorescence lecture. You can see that in this schematic, this photomultiplier tube here is right in line with the direct thing here. So this will be looking at the scattering of the light, while the light that's, they should have like a mirror over here as well. You can see that it's detecting fluorescence. So this is forward scattering. I can spell. And then this is like side scattering. And depending on how complex your flow cytometer is, you can detect different wavelengths. We'll talk about that in the breakout groups of like how many spectral channels there are. And then the common representation of the data, you can see here that there's forward scattering and side scattering. You can make plots of they're two-dimensional plots based on two signatures. So you can do, with all the different combinations of the channels that you have, you can plot scattering versus each other. You can have, here you can see that this is two different fluorescent dyes. So you have a FITC dye here, common dye, PE, I'm not sure which it stands for. So these are common ways of representing the data where each individual point is plotted over here represents a single cell. So you're going to get lots of data, millions of data points, and then this is represented in a contour plot. Again, here, each of these individual data points is an individual cell that was detected and the intensity plotted. So you can note that this is on log scales. But after plotting all of that, then data analysis is a big component of flow cytometry. They term it trying to organize different groups, identifying what those groups mean as gating. And there's a lot of work on statistical analysis of like, how can you decide what are different populations within this data. You can see that there's quite a bit of overlap. Sometimes it might not be as clearly defined with gaps between here. You can see in this example, they're showing different cell types have different amounts of scattering, since scattering will depend on the size of the cell that's present there. So any questions about the basics of flow cytometry? Okay. So flow cytometry can also be used in combination with, okay, this is just getting data out about counting the cells, but then if you want to use those cells after they've been sorted or after they've been identified to be within an individual population, you can then hook this up with a cell sorter. So this would be the output of your flow cytometer. So this is after, so up here would be like your optical laser and your readout. And instead of just putting this into a waste container, you can have a cell sorter attached at the end there. So based on the subtype or epitope of expression, you can sort the cells. So you need to know something about your cells, that it's going to be user defined criteria based on the scattering or fluorescence signal. To identify which, what your sort criteria is. So very quickly after, I guess the laser, I guess in the schematic, sorry, the laser interrogation is right there. So apologize for that. So based on something that you provide, the user provides, after it passes through and with the timing and with fast computer control, it'll say, okay, if we say this is 1, 2, and 3, the cell follows the criteria for group 1, let's say has high side scattering and low forward scattering. We want to sort out the granulocyte cells over here. Then with the computer control, the cells are at this point, you can see this break off point, that at the exit of the flow cytometer, you're going to have a droplet with the cell of interest, break off from the stream, the liquid stream up here. And to achieve this break off, the nozzle at the end here is going to be vibrating at a very high frequency. So this high frequency vibration will cause droplets to form, and it can do so at a very fast rate. If you're remembering, like, we're analyzing millions of cells. So if you want to do this in a reasonable amount of time, they're going to, with that high frequency, you can make 30 to 100,000, let's say, kilo droplets per second. So this is a very fast technique with that. And at this point, the cell sorter, the key thing here is that there's these voltage plates to create an electric field in this area. And based on your cell sorting criteria, that will then tell you what voltage, applied voltage you use. So that applied voltage strength will then, as the droplet is in this phase over here, suspended in air, it will then direct it to a different collection vial at the bottom there. So some of this direction reminds me of mass spec, where you're trying to, based on the mass to charge ratio, it'll direct those charges when we're talking about like time of flight or the different detectors in mass spec. Okay, so any questions with the cell sorting? Okay. So we'll move into the breakout groups now. Let me pause recording and share my screen. In interest of time, I'm just going to use, I'm not going to write text, but I do want to mention a couple of other cell level techniques other than just flow cytometry. One is traction force microscopy, looking at forces that cells exert. And Austin is, I think that's his topic for his presentation at the end of the semester. So we'll hear more then. But the idea is if you're culturing your cells onto a gel substrate, like what we talked on Tuesday, if you immobilize fluorescent markers, fluorescent beads within that gel, that when the cell exerts forces, pulls on that gel, you can look at the displacement of those beads. So you can see here the pre and post, these colors represent where the beads started off, and then they shifted over after the cell exerts force on it. So this is another like heat map of displacement of beads, where this axis here will be, I'm guessing, yeah, this is in microns. So how far the beads are moving. So then these red areas are where there's more force, just more displacement. And then you can use equations going from displacement to actually units of the forces in pascals here. An alternative to traction force microscopy, which is gels with beads, is you can also have nanopillar arrays. So if you use nanofabrication or microfabrication techniques, you can look at these flexible pillars that then look at the displacement of those as cells exert forces. Also looking at, so this is cells exerting forces on a substrate below them, but there's also looking at cell-cell forces as well. So one way of doing this is with a micropipette assay, where you have a cell of interest and typically a red blood cell or an RBC, because it has a very deformable membrane. And your cell of interest will have, and your red blood cell will have certain cell adhesion proteins that will then, you bring them into contact, you leave them in contact, and then you exert force and pull the cells apart from one another. And looking at the time before the cells actually de-adhere from one another will give you information about the kinetics for that disruption, which you can then relate to the energetics of the cell-to-cell adhesion. So that's another force-based technique. And then I just wanted to point out that, like I mentioned, that there's a lot of microscopy and imaging and fluorescence or bright field or dark field, all the microscopy that we explored earlier in the semester applied to cells. And there's a lot of work. Typically people perform an image J of looking at, can you identify the number of cells, the size, the shape, the brightness. So going through binarization of the images and segmentation to then count how many cells there are or track those cells with location. So this figure here is from some work from one of the groups I was in, my postdoc, Martin Grubel's group, and Jan Chemla at UIUC. And here they tracked many different bacteria and looked at them running and then tumbling are these areas where the bacteria stays in one spot. So that relates to when I was talking about the different flagella and bacteria, and they were looking at how these flagella and their activity respond to different, respond to light with different photoreceptor proteins present near the flagella. So ideas that we've talked about at the single molecule level and microscopy also apply at the cellular level as well. And also in terms of like this image analysis, I do microscopy, I do image processing, so I prefer this way, but also recognize that if you're in a more bio-focused lab, they might rely on some older techniques of, they literally sell these guys cell counting by hand, where you're looking through the microscope and you see that there's these two things. You put your thumb, you count how many cells you see that are alive versus dead. There's this technique here that was developed in the 1800s of depositing, I think it was in the 1800s, late 1800s or early 1900s, from a French scientist, of having this grid and you deposit your sample, and depending on like how many cells you count within the grid, you can then do mathematics to scale this up to count what the density of cells are versus, and then there's also black box systems that you'll see where you introduce a solution and it'll do the counting for you. So if you're not collecting the images on the microscope yourself, these are some alternative methods you may come across of looking at cells. And of course, like I mentioned at the start, many of the other methods that we've used have been applied in cell regions, like when we talked about FRET, fluorescence resonance energy transfer, to look at conformation of individual proteins or nucleic acids or the like. This is an example of membrane bound protein, and depending on different stimuli, and here this is in a signaling pathway, that you can report on the conformational change with the FRET-based sensor, where instead of looking at the molecular level or doing this in a fluorimeter, you're doing it on a microscope and looking at the individual cell. So here you can see the cyan fluorescent protein and yellow fluorescent protein here, that is based on if it's in a lipid raft region versus non-lipid raft region, you'll either see high acceptor or high donor intensities. So you can see that you can then plot this spatially within the cell and get an idea of what conformation, also what area of the membrane, if it's in a lipid raft or non-raft region. And then similarly, I mentioned like mass spectrometry, you can do this cell by cell, where just like the cell sorter, you can then hook up your flow cytometer instead of going to that electrostatic plate to then sort the cells into different vials. You can just have the single cells be fed into a mass spec and then perform mass spectrometry cell by cell. So that's another example of a technique we've previously discussed being applied at the cellular level instead of the molecular level. So that's why I wanted to share today. Any questions, comments? Cool. Well, thanks for your work in the breakout sessions to get into the details of flow cytometry, and we'll see you guys on Tuesday.